All right, we're at a minute after 3, so we're going to go ahead and get started. And so our next presenter is Shari Dutton, who is an Extension Associate for Beginning Farmers at the University of Kentucky. And her presentation will be on growing plants for natural dye use. All right.
Hi guys. Oh, I get the clicker. Okay. So this is a culmination of 4 years so far. We are anticipating going forward with this in years to come of work. And big button. Let's advance it. So you can't hear me. I can scream without this mic, but you do. She's had experience. Yeah, I'm used to teaching over the sound of helicopters and trains and ambulances on UK's campus. So I can really, I can be quite loud. And so if I'm too loud, please tell me. This project I did not do alone. I want to let you know about Crystal Gregory. She's an artist, researcher, and endowed professor in fiber in the School of Art and Visual Studies at UK. And then Dr. Ann Frances Miller is a professor in the Department of Chemistry. So our funding for this project came through with the Sustainable Color for Kentucky's Future project that is basically culminated and is ending in December, and is funded by the Office of Sustainability through the Challenge Grant. So, as you well know, horticulture uses plants through cultivation to reap economic and aesthetic rewards. And our reward today to talk about is textile color for textiles. So, in this talk, we'll focus on the plant type, plant names. If you're curious about Latin, we might cover a little bit of that. Parts of plants that are important for natural dye, production, harvesting, and dye outcomes. So you're going to see a little bit of color come out of this. So the types of plants we're going to concentrate on today are going to be specifically herbs, okay? So soft, non-woody, seed-bearing plants with self-supporting stems. And we'll talk a little bit about all three, annuals, biennials, and perennials. Okay, so annual plants, a lot of you may already know this, but it completes its entire life cycle during a single growing season. In cultivation, it's considered a short-term resident. And the lowly marigold, which is not going to be so lowly in this presentation, is an annual. So frequent harvesting of annual flowers, such as we would do if we were harvesting for natural dye processing, can actually increase our harvest length. And so frequent harvesting is before full development of the seeds, and that's actually to your benefit. In development and harvest length. The biennial plant, it's important to appreciate this life cycle because it's going to complete its life cycle during 2 growing seasons. Biennials persist in the garden through the winter, and dye plants, woad and weld, are both biennials. Why this is important to appreciate is because once you get this vernalization period, when it goes through the winter time, the biennial plant then changes its physiology and it begins to initiate a bloom spike from the rosetted foliage. The foliage is what produces your dye for woad. And the dye properties, because of the physiology change, it's now focusing on flowering and fruiting, the dye properties are greatly reduced at that point. So if you've got biennials, know what part you're harvesting and be aware that flowering could reduce the quality of your dye outcome. And then, of course, perennials, they grow year after year. Cultivation's considered a long-time resident in our black-eyed Susan, Rudbeckia fulgida, or Rudbeckia hirta. Both can be used as dye plants. are perennial plants. This is important because if you start a production system that uses perennial plants, you need to plan for weed invasion, both woody invasives and herbaceous invasives, and that competition will harm your overall harvest production amount. So perennial plant systems are a little bit different than annuals because they're long-term. Okay, and another point I want to bring out about black-eyed Susan and some others is that frequent harvesting of young flowers, or those flowers that still have active disc flowers on black-eyed Susan, or the brown part, or the black part. There are many, many flowers, that 200,000 or 200 million that you mentioned. You can get that number pretty quick with a heavy population of disc-flowered plants. I think I just got louder. So anyway, yes, frequent harvesting can increase your yield over time through the season. It's good for dyers. So plant parts that are important to the natural dyer are going to be your aerial portions, flowers, of course, leaves, stems, fruit, and seeds. And then also underground portions. So madder root is up here in the right-hand corner, and it looks kind of like a mesh of earthworms, but that's actually roots. And then, surprisingly enough, a sunflower, this particular one is the Hopi black dye sunflower, you use the seed. So you look for fully developed seed with color, and you harvest that head. And we'll get to that here in a second. Cultivating plants allows us to choose the best species or variety for reproducible color. So I'm a horticulturist, and this is kind of something I have not seen in the literature, is a mention of, varieties for specific tonal variation and dye outcome, and I think it's really exciting. And I think you guys could take this home and run with it. And hopefully by the end of this talk, you'll feel empowered to do that. But we have a lot of plant breeding and cultivars out there. And it could be some really exciting dye outcomes unique to your farm or to your operation if you just do a little bit of research and development on it. And I'll show you why. So our palette for this project was Hopi sunflower, gives us grays, Seneca red stalker corn, gray purples. We got a student right now in the class that is creating royal purple from husks of Seneca red stalker corn. It was incredible. She did it yesterday in the lab. French marigold, Tagetes patula, Bonanza bolero is the one we're going to focus on in this talk. Cosmos tango, that is an heirloom variety that is very, very, very much a giver of color, bright orange. Black-eyed Susan, browns and tans. Dyer's Coreopsis, Coreopsis tinctoria of yellows and oranges. Okay, here, yeah. If I can figure it out. One more. There you go. We'll go over each one of these specifically, and I brought the swatch book. So if anybody wants to look at the real deal, we actually, this class that is ongoing that was funded through the sustainability funding, we are teaching a class. And so we finished these swatches last week. So this really is hot off the press. It's I'm pretty excited about it. If you can't tell, I am. So we're going to talk about plant names. We're going to look a little bit of Latin, just because it's important. It's going to give you an edge on that reproducibility, okay? So plants are related, just like we are, to each other, and have special names to identify them. And our marigold has a lot of names. So yeah, Stinking Roger. I didn't know about that, but somehow Wikipedia did. Khaki weed, pot marigold, Indian marigold, French marigold, African marigold, or marigold. Now, what in the world? Which is it? Well, it happens to be the common name in our area. A lot of these common names are common in other areas of the world, but in our area, the word for this is French marigold. So French marigold is a smaller marigold in stature and in petal number. And it has another name, has a scientific name with a genus and a specific epithet of Tagetes patula. So if we get a really interesting dye outcome from a French marigold, we're going to want to know what to buy next year, right? So we have lots of horticultural selections called cultivars. And cultivar names are fancy, and they're not Latin. That just means they're within the language that was developed in. So it's a fancy name, set off by single quotes and not italicized. So this marigold then becomes Tagetes patula Bonanza Bolero, or French marigold Bonanza Bolero. Okay. Now we know what to get if we really like our dye outcome. And why is that important? So it has vibrant, long-lasting blooms. It's an incredible giver of color, a reproducible color. It doesn't take very many flowers. Weight of fiber to weight of plant material. Dried is 100%, so however many grams of fiber you're going to dye, that's how many grams of dried petals you would use, which you don't have to take the petals away from the calyx of the flower. You simply pull the flower and dry it. We'll talk about that here in a second. This is an All-America Selection winner, so it is not sensitive to short or long days. It has a uniform growth habit. You know how tall it's going to get, you know how wide it's going to get, and you know how to space it out for best performance. And there are more blooms per plant without pinching, so you don't have to coax it to produce more. It is bred to do that, so you don't have to do anything special. So it's good to know what to expect. We have plant production factors that help us produce a good crop for color. And we'll just basically touch on that. One being environment of the soil, environment of radiant energy or light quality, and another being the environment considering air. And it's not air you breathe, it's air in the roots zone. So soil is a reservoir of organic and inorganic components. It's a dynamic system, an intimate association. That soil provides mechanical support of the plant, but modifications to that matrix can greatly affect dye outcomes. So acidity of the soil or basic properties of the soil, alkalinity of your water, things that are interacting with the plant can affect your dye outcome, which means that different parts of Kentucky might actually produce a little bit different color palette, which might make your farm or your area unique from someone else, and could be advantageous. Not a detriment, but an advantage. So air, good plant development depends with good air movement, and we're talking about that relative to the soil. And so typically very wet soils have low oxygen and high carbon dioxide, and for dye plants, that's typically not the environment you want them in, and so siding of most plants grown for natural dye use should not be in soils that can become waterlogged, as a general rule. And then radiant energy. I hear a lot of people say, I get full sun, and the stress here on this slide is, okay, what is your full sun? Dye plants usually need abundant light energy, so that's not 8 a.m. to 8 a.m. to 11 a.m. That's basically 8 a.m. to 5 or 6 p.m. That's all the sun they can get as long as they can get it, with little to no interference. We learned that when we planted 2 gardens at the Arboretum in Lexington. We had one garden in a partially shaded location and another one in a full sun location, and yield and quality was dramatically different between the two. So just to sum that section up, the best sites was a good site, good soil tilth and adequate organic components, adequate uniform soil moisture that drains well, and a site that receives full sun. Now, I want to talk a little bit about starting plants, and I got a handout on the tables. Looks like this. It's a Master Gardener printout on seed viability. I wanted to point it out first so I don't forget it mainly. But we're going to talk a little bit about heirloom seed and named cultivar seed, because those are two general categories, and both are important if you want to try natural dyeing. So heirloom seed is seed passed down from generation to generation within a community. It can be, culturally very important. They're open pollinated. Once the plants cross pollinate, those progeny display varied traits. We're gonna look at a little pro and con of that, because there is a pro and there is a con. Saved seed must reach physiological maturity. Hopi black dye sunflower, the Seneca red stalker corn, Dyer's coreopsis, and Tango cosmos are all heirloom species. And we're going to look at how beautifully they yield color. Okay, so our hybrid varieties, on the other hand, are more expensive to purchase because of the time and the labor it takes to do the crosses. But you have uniform, predictable performance. Now, we had 3 interns this summer. They worked in hot, humid conditions. And the marigolds that they worked with, that were all the same size and all the same height, allowed them to put together a little harvest protocol to ease their hot, humid pain. And I took them over to another plot that had marigolds of different sizes, and some that fell over and some that didn't bloom as well. And I said, well, let's go collect from these. And they were like, no, I can't pull my wagon up to that, and harvest that quickly. I'm hot and I'm tired. So they didn't want anything to do with that. They wanted the hybrid varieties where every plant was pretty much the same height, same width, and the same abundance of flowers that they could harvest. So, 20-year-olds will let you know real quickly if they don't like the system you've put in place. So seeds saved from a hybrid population usually are not used commercially because the progeny will display a great variability of plant performance. And so some will be tall, some will be short, some won't have very many blooms on them, some may split or lodge easily in the wind. They have all kinds of performance attitudes. So the germination process, and this is really either category, hybrid or heirloom. Your seed must be viable, it must be alive. The seed must have an appropriate environment to germinate and to grow. And so that little handout there is a quick, kind of a quick and dirty way of seeing that the seed, whether heirloom or hybrid, maybe you've kept your seed for a while and you want to know if it's still alive. That little sheet will give you kind of a Q&D way of telling. And then you can make the decision of whether to order more seed or waste the real estate, to try and grow a crop if it has poor germability. So, and if you don't have one of those, let me know and I'll get you one. Okay, so this is the URL that page came from. So that was just on there. But you guys have the hard copy, so I would advise you to test your seeds before you sow. No matter if you're collecting those seeds yourself or you have some that you've bought and they're a couple years old, I would still test them. So this is kind of a small scale dye plant production, quick thoughts, right? You need clean, viable seed. You need clean, peat-based growing media, appropriate, clean seed starting trays and materials, and guides are great. So UK's got several guides. Master gardeners have done great jobs in producing videos and information on that. Certainly can check that out. Grand Prismatic Seed is a seed company that we used to buy the seed for some of this, and so they have a really great like full color dye garden, seed starting dye garden tutorial. I thought that might be helpful for you. Johnny's Seeds has great full color, nice seed starting kits, full color tutorials. So those are just two that I put in there. It's I'm not advertising for these guys. It's not an endorsement. It's just I thought they were good resources. But there's a lot of resources out there. Definitely, anytime you want a resource, typing in .edu is your friend, right? These are just two companies that I've worked with, and their guides are good as well. So seed suppliers. So these were our suppliers. So Grand Prismatic Seed, they were the sellers for Tango, Cosmos, Dyer's Coreopsis, the black sunflower. Parks, our French marigold, Bonanza Bolero. Holmes Seed Company was the red stalker corn. And Prairie Moon, among other things, they have black-eyed Susan. They have lots and lots of native plants. So. No, that's the seed company. So there's no variety. It's a straight species, Rudbeckia fulgida. Rudbeckias Rudbeckia hirta gives different dye results from Rudbeckia fulgida. So it will work, but you'll get different colors. And leaves give different colors than flowers. But Prairie Moon, Holmes Seed, and Park Seed and Grand Prismatic are all seed companies. Yeah. So this is our dye garden in 2022. The dye garden then featured native and non-native plants. And it produced a variety of natural plant colors for textiles. Blues, greens, yellows, reds, and pinks came from I think it was 35. There were 35 different plants within the dye garden area that produced these colors. So our plants were grown as 50-cell plug plants. And so that's, when I say a plug plant, it's represented there in the top corner. You've got a little cache of soil with a good root system, and you're pulling that out and you're putting that, into the ground in a production manner. Our secret sauce for growing our plants was a slow-release granular fertilizer with an 18-5-12 NPK. Here's the real secret: 5 to 6 month release. If you get a 3-month release and you go into July, August, you'll have no fertilizer left by the time you reach the 1st of August, because it's over, it's done. The granular fertilizer has released all it has to release. So you need one that has a little bit longer release period to keep things going, so that you don't have to put another input in later in the year. So we watered the plants in, we mulch with a hardwood mulch. Our biggest challenge was not fertility, because we'd covered that. We did have supplemental water when it was needed. Our biggest challenge was weeding. Okay, so lots and lots of weed pressure, even though we had the hardwood mulch, and our interns greatly complained about the weeding tasks. So it was vital, though, for good harvest. So this was Allie's method. She would pull her wagon up and open the bags, and in would go the petals, and then they would wheel them to the next area. They made their harvests early mornings. They were out there before the rooster crowed, and because it was so hot, and Mondays and Wednesdays, and they collected fully open young flowers. Okay, so that's kind of important. Note, fully open and young. They're separated out in a post-harvest treatments according to variety and species, and they were separated for freezing, a post-harvest treatment of freezing, and for open air drying. And we have several open air drying techniques to share with you today. In the Ziploc bags, they were in Ziploc bags because of the freezing. So anything that was air dried was collected in paper bags. All right, so harvesting. How am I doing on time? Am I doing okay? Oh, okay, so harvesting, we did this in various ways. These are herb racks. They were hanging in this tunnel. It was an unused tunnel. And the herb racks allowed for adequate airflow around the petals, fairly quick dry times. We used the benches with some screening to dry our sunflowers. The con for the drying rack, the herb drying racks, is that UV light degrades some of the dye properties of the petals. And so we actually employed the herb drying racks very late in the season in September when our light quality started to wane. We'll look at that here in a second. Again, we had three post-harvest air drying, actually 4 post-harvest air drying systems. One was a forced air oven, similar to what you would get if you had a dehydrator. We dried in that. We have plants that are on the bench and covered by regular window screening, right? We had petals and plant debris that was covered with 90% shade cloth, and this is horticultural grade shade cloth that's used in the industry. So you would get that to shade a greenhouse or a hoop house. So guaranteed 90%. And then the black is 99.9%. Woo, sound like a kitchen problem. 99.9% blackout cloth. That was woven, and it allowed air circulation, but not adequate air circulation. So those petals that were under that were actually slower to dry, and we actually worried about degradation from, senescence, slow senescence. So when we folded the material over, so basically just formed a taco, then this next picture shows that, and the blackout fabric then had a white side to reflect, radiant heat buildup under there. But still, we were worried about the length of time it took to dry under there because it wasn't adequate air circulation. The best results that we got for a dye quality is coming from our 90% shade cloth. The window screening allowed a little bit too much UV degradation. So what that translates into is it's going to take more petals. So that 100% weight of fiber to weight of plant, you would need to increase your weight of plant material to make up for the degradation from the UV light. So, okay. How do you harvest? So it matters. We would pull upwards on the calyx of the plant and basically pop up the flower off of the plant, and that kept us from damaging the petals. We didn't want any necrosis, being introduced. We wanted to get them frozen or dried as quickly as possible. We did not harvest declining flowers, so there's not any necrotic material in the mix. And for marigold and cosmos, you can use it fresh, dried, or frozen. Now, using it fresh, you have to increase your weight of plant material, because at least 80% of that weight is water, right? So you're better to, when you use it fresh, to go 200% weight of plant to weight of fiber. But you're out there harvesting. I don't know, dyeing and harvesting is kind of hard anyway. I'd want to save it to the winter. So what do you get? You get this. There's three types of natural fibers here. The top is going to always be silk, and underneath the silk is a wool fabric swatch, and underneath the wool is a cotton fabric swatch. Now, I brought my book. It's really hard to show the variety of how each fiber takes the same dye bath, but you're welcome to have a look at my book. I just sort of concentrated on silk just because that was easier for the talk. But the silk fabric is scoured first, and that's to remove sericin, which is a gum that exists on silk. So it's scoured. It's scoured in a pH-neutral detergent like Dawn. Dawn dishwashing soap is one of them. We mordanted the silk fabric with aluminum sulfate or alum. You may have heard reference to alum. That's what that is. We used 100% weight of fiber to dry plant material, and the control swatch is always going to be up here in this left corner on my slides, and there's no modifications on the control. It was just put into the dye bath, and we'll talk about how that bath was made here in a second. So the rest of these swatches are products of modifiers, and so we took that same result from the dye bath, and we placed fabric swatches in an iron-modified environment, some swatches in an acid-modified environment, and that acid would be citric acid, a soda ash modified environment, and that would be an alkaline treatment, in indigo overdye. And so we had an indigo vat that the students prepared for our overdye experiments. So that's marigold. So each one of these is going to be the same way of explaining what you're seeing. And that is our cosmos. Cosmos is an excellent giver of color. Those are those oranges again. You overdye that with indigo, and you get really a nice rust brown from iron. So you've got quite a bit of a color array there just with cosmos and a few modifiers. The second dip, there's a couple slides in here that represent a second dip, and we were trying to see if we could intensify the results, and really that didn't happen. I mean, you got good color, so there's, it's kind of like you can't improve on it really. And harvesting Rudbeckia, basically the same way, except that your pollinators tend to tell you what disc flowers, if there are disc flowers that are still young, right? And so sometimes for Rudbeckia hirta anyway, this is hirta autumn colors. For Rudbeckia hirta, the ray flowers are very colorful, even though the flower is in decline or it's old. And so the interns would look for signs of pollinator activity, and that would be the flowers they would collect for the post-harvest treatments. And so a soldier beetle is a good indicator. He was busy. Sorry, it's a little, wind must have moved the flower, but I had good intentions. But he's busy on there, and you just pull the flower upwards from the stem, similar to cosmos and marigold, and you can use this fresh or dried. And like I said, the Rudbeckia varieties, cross, like say Rudbeckia hirta Prairie Sun has disc flowers that are green, and so the dye outcome from that will be different from Rudbeckia autumn colors, because so many disc flowers are such a different color, right? And it really does make a difference. So you could concentrate just on Rudbeckia and get a color array just from that. So my swatch book represents Rudbeckia fulgida, not hirta. So this is the straight species of black-eyed Susan. And in the swatch book, you'll see color representation of leaves and color representation of flowers. And I just put flowers up here. But nice array of colors from that. And, you can leave some for the pollinators. Rudbeckia fulgida is so giving of flowers that, you can leave some for the pollinators while you're harvesting for your color. Our Hopi black-dyed sunflower, so it has kind of some special needs. It has flower heads that when they're fully fertilized, the capitulum, the head, starts to bend down. That's telling you you've got all the flowers pollinated. You need to bag it with a tool bag, something that allows air movement, because you don't want a transpirational sweat to build up and start to rot that head. So you need to use something that allows air movement, and we use tulle. And that keeps the bird activity down. And the heads are removed basically to cure on the bench when you start to see kind of brown bracts, yellow-brown bracts. And certainly watch for rodent activity, because they know what food is just like anything else. And you can harvest your dry seeds out of these heads, placing them in glass jars with a desiccant packet or several desiccant packets if you feel the need to go with a couple. Give you a reason to reuse those desiccant packets you've got stashed in a drawer. So that's how we did that, and that's what we got. So some nice grays, and the over dye, I think, since you've got that kind of that almost a tannin type background, you got a nice indigo over dye with that because of that underlying hue. Dyer's coreopsis, this is a small flower. It's about the size of a dime. And the heirloom variety on this meant that we got many, many colors. So we did not separate the colors to see what the dye outcomes would be of different segments of the population. We just cut the stems and leaves and flowers and put them all in a bag and dried them. And that's what we got. So pretty nice color out of that. Yeah. All the interns, they wanted to mutiny when I said, maybe we should just get the flowers, and they're like, oh no, I'm not. Yeah, just the flower only. They refused. They were like, we aren't getting paid enough. So yeah, so we used everything and still got great color. But that's a mention of the heirloom variation. If you've got small real estate, and we'll get to here's the mention about the small real estate. This Seneca red stalker corn, we want to harvest shucks that have this deep purple coloration, or leaf sheaths that have deep purple coloration. Now, of the population of plants that was in our plot, only a certain percentage exhibited this deep purple coloration. If you've got just a little bit of real estate to devote to this, an heirloom, some of that real estate won't yield you very good results, because it's the whole range of possibility, right? It's open pollinated, and it's not gonna exhibit reproducible color in every plant. So mention of the stalk. So yeah, so our first question, I was like, we get in the dye lab, and I'm like, well, it's called Seneca Red Stalker. Let's just do the stalk. So we pulled every leaf, every little bit of husk, every little leaf sheath, every bit of it, cut up the stalks. Oh, terrible, terrible, terrible. It was just this, well, it was the color of the paint up there. It's what's in the notebook. You can see it. No color, no color. So, but the sheaths, the leaf sheaths and the husks is where your cell density is in the plant and where the density of the color is. And that's what we got. So we've got kind of this purple gray, and then a nice olive for the alkaline modified, and kind of a deeper olive with an indigo overdye. So nice results. And this one, a student her results on Monday from this was yielding a royal purple. And so she's going to have to divulge her secrets to get her grades. So we all want to know what she did to treat it. But once she learned about these modifiers, then she decided she's a chemist. She's a chemistry student. Once she learned about the modifiers, she went digging into the literature and came up, I think, with some pretty interesting treatments. So I'll share that at a later date once I find out what she did. So plants produced a colored dye bath. So what we did was we would take our plant material, we would put that plant material through a long, gentle simmer, temperature no higher than 180 degrees Fahrenheit. And we would do that for about an hour. We just look at the dye bath and see if we had good color from it, and make a judgment call. We would strain that dye bath, just through a regular kitchen strainer, retain that liquid, put it back into the pot, take it back up to 140 to 180 degrees Fahrenheit, and then put our material in, our scoured, mordanted material. And that was all there was to it. This is a representation. Actually, this is from Berea, the festival that they have in Berea. But we did that. We did this on an induction hot plate, and just with stainless steel pots. This is just kind of a folksy representation from long ago. And this is just a summary of what the color we just saw. Just kind of give you an idea of those 6 plants. And this is a QR code that will take you to the Arboretum's Dye Garden. And the Dye Garden, this site basically gives you some ways to start to do your own natural dyeing. It's going to talk about mordanting. It's going to talk about how to make a dye bath, how to mordant. What is it? How do I do it? What was that again? What'd you use? It's on there. One of our interns, Bailey, designed this part of the Arboretum's website, Dye Garden website, and put this information in for you. There's like like the 4 little bars, and you click on that, and you get a drop-down menu. So the what alls and what ifs are under there, and then there are plant descriptions as well. So that's something you can explore when you go home. Yeah, and that's about it. How did I do? I'm going to do questions? Okay, yeah.
I know you probably know Lori Bowling.
Yeah.
County.
Yeah.
She had me to do freeze dry.
Oh, you did the freeze-dried?
Yes.
Yeah, cool.
Has anyone
No.
used those yet?
No, she hasn't sent those to me yet. But I'm sure she will. But yeah, no, I knew somebody was doing that, and that was the one treatment we didn't. Oh, I'm glad to know that. Okay, cool. Yeah, what the results were. Well, hey. Oh, did you? Well, cool. Yeah, I mean, this is really fun. And my hope is that this can get taken to your farms. And once you dye fabric, scoured and mordanted fabric, once you dye it, now it's a shelf-stable product. That can go through Etsy. That can go to rug hookers. That can go to knitters. That can be yarn. That can be yardage of fabric, silk, wool, cotton. I mean, it's a great way to diversify using a production system that you probably already use for cut flowers. So, very little input. Anything else? No? Well, okay, if you need me, I'm at the University of Kentucky. I'm in the Department of Horticulture, and just reach out, right?
Okay, so we are done with everything here at the farm for today. So the next thing on the agenda will be the banquet at the hotel at 6. The last I heard, the buses should be getting here around 4:15 to take you all back to the hotel. Somebody did ask earlier about if these presentations will be available, and the answer is yes. I do not know exactly when, but they will be posted on the KYSU Ag, just KYSU AG YouTube page, and I believe will also be sent out in an email afterwards. Yes, and thank you all for being here today. Yeah, I think that's it.
It would be something to possibly look into.
One last thing I forgot to mention that's super important. If you could fill out that survey, it really, really helps us to keep programs like this going and to make sure we're having an impact on you all. So if you could please complete that day 2 conference survey, it would be greatly appreciated.